Hello everyone. Today we'll explore genetic architecture, focusing on the structure of nucleic acid, RNA, and DNA. We will learn how genetic information is stored and transferred in living organisms. Okay, let's start with the nitrogenous bases. That is the fundamental building blocks, right? So both DNA and RNA are constructed using specific nitrogenous bases. Okay. So there are five important nitrogenous bases found in nucleic acid: adenine, guanine, cytosine, thymine, and uracil. Okay. So adenine pairs with the thymine in DNA, while in RNA it pairs with the uracil. Okay. So thymine is only found in DNA and uracil is only found in RNA. So this is exclusive for DNA and this is exclusive for RNA. And adenine, cytosine, and guanine are universal. It is found both in DNA and the RNA. Okay. So cytosine always pairs with the guanine. So in DNA, adenine pairs with the thymine. And in RNA, adenine pairs with the uracil. Okay. And cytosine always pairs with the guanine. Okay. So these complementary base pairs form the basis of the genetic code. Okay. Now let's see the difference between DNA and the RNA bases. Okay. So the DNA base pairing books. So thymine is only found in DNA, right? Thymine is only found in DNA, whereas uracil is only found in RNA. So this difference helps distinguish DNA from RNA and support their different biological functions. So as we mentioned, the adenine locks with the thymine and the cytosine locks with the guanine. And in RNA, adenine locks with the uracil and the cytosine locks with the guanine. Okay, so this strict complementary base pairing logic is the foundational mechanism for genetic storage and transfer. Got it? Now let's see RNA polymer. So RNA is a polymer formed by joining nucleotide monomers through condensation reactions. RNA contains ribose sugar and plays a major role in protein synthesis, right? So let's see. So here you can see the assembly, right? RNA is formed when individual nucleotide monomers are joined together by condensation reactions. So this is the foundation, right? Built on a structural backbone, ribose sugar. And what is the form factor? Functions typically as a flexible single-stranded molecules, right? So as we have mentioned, it is formed by joining nucleotide monomers through, these are the nucleotide monomers, right? Each one is a monomer and they are tied together. Okay. And they contains ribose sugar and plays a major role in the protein synthesis. Got it? Now let's check the types of RNA and the four specialized roles. So there are three main type of RNA. First one is messenger RNA, that is mRNA. And it carries genetic instruction from DNA. So that is why it is also called the courier. It carries genetic information directly from DNA to the rest of the cell. Second one is transfer RNA or tRNA. Okay. So they transport amino acid to the ribosome. So they transport specific amino acid required for protein synthesis. That is why it is called a transporter. Then we have the ribosomal RNA or rRNA. So they form part of the ribosome and help catalyze protein synthesis. So that is why they are called the factory flow. Forms the part of ribosomes and physically help in making the proteins. Okay. Then next we have the ATP, the energy battery. So adenosine triphosphate or ATP is a single nucleotide that stores and transfers chemical energy in cells. It powers many cellular activities, right? So it is a single specialized nucleotide utilized specifically to store chemical energy. So these are the four specialized roles of RNA. Okay. Now let's see the double helix architecture of DNA. So DNA has a double helix structure made of two antiparallel strands. Okay, so the strands are antiparallel. The sugar phosphate backbone forms the outside of the helix, while the nitrogenous bases are stacked inside. So as you can see, the outside layers of the double helix model are made up of sugar phosphate bond, right? The sugar and phosphate bond we can see here. And the inside stacks are the nitrogenous bases, right? The adenine, guanine, thymine, all these things. Okay. They help maintaining the stability of the DNA molecule. So the exterior scaffolding is by the sugar phosphate backbone sit on the outside of the helix, forming a protective railing. And in the internal data storage, there's the nitrogenous bases, right? They are housed safely inside the helix and protected from the external cellular environment. Here. So now let's see the shape of double strand in detail. So as you can see here, the complementary base pairing. In DNA, adenine pairs with the thymine. Okay, adenine pairs with the thymine and guanine pairs with the cytosine, right? So these complementary pairs are held together by hydrogen bonds, helping maintain the stability of the DNA molecule. So you can see the anti-parallel strands here and the hydrogen bonds. So the structure contains two nucleotide strands running in physically opposite direction. That is why it is called anti-parallel, ensuring maximum structural stability. So one side will be 5' and carbon and other side will be 3' and for the next strand it will be the just opposite, right? 3' and 5' and like this. Okay. And inside you can see the all the nucleotides and all. Okay. Clear? And the hydrogen bond, the two antiparallel strands are firmly held together by delicate hydrogen bond forming strictly between the complementary base pair. Like we mentioned, A pairs with T, G pairs with the C. Okay. So, as you can see, the DNA versus RNA architecture. So let's compare the DNA and the RNA system. Okay. So you can see the structural form. DNA is double helix model, but RNA is single polymer chain. And what is the sugar backbone? Deoxyribose. In the RNA, it is just ribose. And what is the base composition? It contains adenine, cytosine, guanine, and thymine. But in RNA, it will be adenine, cytosine, guanine, and uracil. And what is the complementary pairing? In DNA, A pairs with T, G pairs with C. But in RNA, A pairs with U and G pairs with the C. Okay. So, from the five foundational nitrogen bases, nature engineers stable anti-parallel double helix of DNA to store the master genetic blueprint. Through condensation reaction, nucleotide monomers form the dynamic single-stranded polymer of RNA to transcribe, transport, and execute the blueprint. Together, driven by the chemical energy of ATP, these molecular architectures form a mechanical basis of life. So to conclude, the nucleic acids are essential biological molecules that store and transfer genetic information. Understanding DNA, RNA, and base pairing help us understand how cells function and how traits are inherited. Thanks for watching.